We interrupt this program to bring you a breaking news report from Mayo News.
Good afternoon. We apologize for this interruption, but we have breaking news story to report. According to the PNS Newswire, potential irreversible action is ensuing. It's being reported that there's a major surge in sodium. Do we have anyone at the scene? I'm being told that we're having some technical difficulties. While we're resolving this issue, we're going to bring you some background information. Our technical correspondent here at the station is Amy. What can you tell us about the situation, Amy?
To know what is going on, you first need to have an understanding of the structure of muscle. As an example, I am using a makeshift model to demonstrate skeletal muscle. Imagine that this group of pencils is the large muscle of the shoulder known as the deltoid muscle. You can see that there is a wrapping around the muscle, which is known as the epimysium. Within the muscle are many segregated sections called fascicles. They too are wrapped by a connective tissue called the perimysium. Within each fascicle there are individual muscle fibers represented by the individual pencil. The muscle fiber is also wrapped with a sheath called the endomysium. Lastly, inside of each muscle fiber are numerous myofibrils represented by the pencil lead. Each of these myofibrils is made up of many sarcomeres or muscle segments. The sarcomere is the contracting unit of the muscle cell. Within the sarcomere are thin filaments called actin and thick filaments called myosin. Actin is...
I'm sorry to interrupt, but we're live on scene. Isaiah, are you there?
And now on CSU Eyewitness News at 5, we are near the scene where a person claims to have showed the fastest withdrawal reflexes ever. Witnesses say that the victim/record breaker touched a hot pan being used by a street vendor and showed the fastest withdrawal reflexes ever seen. Here to give us a more in-depth explanation is Dr. Isaac from the CSU Muscle Reflex Research Institute. Dr. Isaac, could you please explain?
Yes, Judith. Thank you for having me here, by the way. Yes, I'm going to give you a brief introduction on how this event occurred. The first thing that happened when this person touched the hot pan, the thermoreceptors at the tip of his fingers were so stimulated that they created an action potential through the afferent neurons that eventually are going to have their axon terminals at the ventral horn of the spinal cord. Two events simultaneously happened when the action potential reached the axon terminal of the afferent neurons. First one is the afferent neurons sent excitatory signals to interneurons, which in turn are going to have their effect on the biceps of the arm. The second event is going to be the afferent neurons are also going to send excitatory signals to inhibitory interneurons that are going to have an effect on the triceps of the arm.
Okay, and for a further explanation, we now go to Aaliyah. Thank you, Dr. Isaac.
Thank you.
The action potential is far ahead of me, and it looks like that the action potential is triggering the release of calcium. And now the calcium is triggering the release of the neurotransmitter acetylcholine, and the acetylcholine is going to diffuse across the cleft. Let's see if I can catch a ride.
Go.
Here we go again. I'm following the action potential racing down the afferent pathway of the motor neuron. I arrived at the terminal button of the neuromuscular junction. The action potential has again stimulated the release of the calcium, and the calcium again has stimulated the release of the acetylcholine. It's everywhere. The vesicles again are moving towards the cleft. I can't go any further. This is Aaliyah, sending it back to the neuromuscular junction.
media, Vincent and Elvira reporting live on the scene where the action potential that was generated from the stimulus and propagated from the afferent to the efferent to the effector muscle excited the muscle. There were a whole chain of events. This is what happened. The active potential opened up voltage-gated calcium channels in the synaptic knob of the efferent neuron. The calcium stimulated acetylcholine release from the vesicles into the synaptic cleft. The acetylcholine binds the receptor in the postsynaptic membrane. Voltage-gated sodium-potassium channels open. Sodium is released into the sarcolemma and potassium leaves. This causes a depolarization, which generates another active potential, which causes excitation of the muscle itself. The active potential goes through the T-tubules, opens up voltage-gated calcium channels in the sarcoplasmic reticulum. Calcium enters into the sarcoplasm. Calcium binds to troponin on the thin filament. Troponin and tropomyosin complex change shape and expose active sites on the actin. Myosin hydrolyzes an ACP molecule which forms ADP and phosphate and activates its coupling position. In this position, it can bind to the actin active sites, which forms actin-myosin cross-bridge. Myosin head releases ADP and phosphate. This action flexes the head to pull the thin filament past thick filament. This is known as the power stroke, and does this again and again by binding more ATP. This forms the contraction. Now it's time to relax. How did it relax, Elvira?
Do you think the work is done? Well, to be able to relax, the muscle needs also ATP, and relaxation happens when calcium is reabsorbed into the sarcoplasmic reticulum. Oh, there it goes. And myosin releases the thin filament. Hey, watch out! Of course, we're in the skeletal muscle, but if this was occurring in the cardiac muscle, the news report would be different. In cardiac muscle, calcium is removed from troponin and sent back into the sarcoplasmic reticulum, but then tropomyosin reblocks the active site, finalizing the contraction relaxation process. So, now you know what's happening with contraction relaxation. This is Elvira Becker reporting from the muscle site. Now let's get back to you, Anchor.
Well, that explains what is happening. Thank you, Elvira and Vincent. It seems like things are finally under control. But what happens when things are not? Let's find out from our crack reporter, Haney. Tell us.
Thank you, Anchor. This is Neuromuscular reporting from When Things Go Wrong Station about skeletal muscle pathology. Skeletal muscles are basically use it or lose it type of muscles, which means that they routinely have to be stimulated in order to contract and also maintain their size and strength. But in the case of atrophy, skeletal muscles diminish in size and strength due to not being stimulated. The common example is that when astronauts were sent to space, they came back with 0.5% reduction on mass weight bearing muscles due to lesser gravity in space and lesser effort for skeletal muscles to move around, which confirms that skeletal muscles are use it or lose it type of muscles. The next skeletal muscle pathology we're going to look at is muscular dystrophy, which is a genetic disease. It's characterized by progressive degeneration of contractile elements of muscle, which eventually will be replaced by fibrous tissue. That's it for me, Neuromuscular. Back to you, Angela.
Thank you, Haney. And in addition to the information that Haney provided, there are also a few neuromuscular junction disorders. The main mechanisms, there would be three. One being altering release of acetylcholine, blocking acetylcholine receptor channels, and preventing inactivation of acetylcholine. As for altering release of acetylcholine, the main example would be the black widow spider venom, which causes explosive release of acetylcholine, which eventually causes death by the diaphragm staying contracted. In the second case, the blocking acetylcholine receptor channel mechanism, there's the autoimmune disease myasthenia gravis, in which the antibodies inactivate acetylcholine receptors, which eventually leads to death by no contraction in the diaphragm. And lastly, the mechanism would be preventing inactivation of acetylcholine. The main example of that would be organophosphates, such as in nerve gas, which irreversibly inhibit acetylcholine esterase, which continually stimulates the muscle because acetylcholine is not reuptaken, which eventually causes death. This concludes our special bulletin. Maya News now returns you to your regularly scheduled program. Thank you.
This has been a breaking news report from Mayo News.